Our technique introduced the egg into the nucleus into the egg. It also activates the oocyte to resume development. In sheep, you can do the two things at the same time. If you do that in the mouse, you will get some egg development, but you will not get offspring. It was shown quite clearly that for the standard procedure, you will only get development in the mouse if you delay activation. So you put the nucleus in and then activate by a different means 2 hours or so later, and you will get a high proportion of offspring. In the sheep, it makes no difference at all. In the cow, there's an advantage in the delay. The mouse, as put in the slide here, it is essential that you delay activation. We have absolutely no idea what accounts for these differences. The rat oocyte is very difficult to work with because it activates spontaneously. Oocytes of many species, I think, will activate spontaneously if you handle them carelessly, if they become old. Various things like that. The rat, it's essentially impossible to stop them, which means that nuclear transfer is very difficult to do and has been very inefficient in that species. There are no clones of primates, despite quite a lot of work in non-human primates. There are no normally developing blastocysts in humans, so there are no human ES cell lines with a ploidy. There are 1 or 2 non-human primate ES cell lines, but literally 1 or 2. It's a very unsuccessful process. I'm going to return to this briefly. The recent publication showing that this is associated with this failure is associated with removing the spindle. If you put another nucleus in and activate that egg so it becomes triploid, it will develop. Normally you can get embryo stem cell lines from that embryo. So if you enucleate it, it's the standard procedure, requires you will not get development. So there's apparently something unknown which is removed, which is essential for normal development of the embryo. The impact of this is illustrated in the comparison between these two species, just mapping broadly the pattern of gene expression at the time of activation in the mouse on the left-hand side and the human on the right-hand side. And the mouse is a comparison of a control, in vitro produced, in vitro fertilized embryos, and nuclear transfer when the nuclei were put into zygotes which were enucleated. And you see in the mouse that there's some differences between these three populations, but really quite small. On the other hand, in the human, this is a transcript of normal embryos donated from vitro fertilization clinics. This is after nuclear transfer if the spindle was removed. And this is a normal embryo in which the inhibitor of transcription, amanitin, has been included. And you can see that there's a great similarity between the amanitin and the nuclear transfer groups, illustrating quite clearly that there's been very little reprogramming of gene expression and the absence of the onset of the transcription from the zygotic genome. That point I mentioned a few minutes ago, there's something being removed which is essential for that critical step of switching on the embryonic genome. We still know remarkably little about the process of reprogramming, what's involved. If you really looked hard through the literature, you might find 2 or 3 papers. This is a relatively new one from John Gurdon's lab, where he showed that nuclear actin is essential for reprogramming of nuclei. So that if you put in an antibody, this is the paler coloured columns, to inhibit the effects of nuclear actin, you did not get expression of the OCT4 gene, whereas if you have a control, it does have an effect and you get significant expression of the OCT4 gene. So he interprets this as indicating that it has a critical role in regulating the chromatin modifications around the OCT4 gene sequences, and so regulating its expression.